Okay, so I'm Dr. Caroline Dalton. I'm a reader in neuroscience and genetics from the Biomolecular Sciences Research Centre, but I'm also theme lead for Living Well with Chronic Disease at the AWRC. So, and I'm going to talk to you about eating behaviours, food cravings, and the influence of age, sex, BMI, and FTO genotype. And I would like to say right from the beginning that I think we started this study and thought we'd find out all of this stuff, and we ended up possibly more confused than when we began. So I just mentioned that from the outset. However, we have published some of this stuff. So, oh, hang on a sec. Oh, yeah. This is the data I'm going to present, or I'm only going to present you a small amount of data just to give you an idea. Is from a PhD student that Dave and I supervised together, which Hannah and Abdullah, who got a PhD just over a year ago now, and in collaboration with Dawn Haddon from my department, and also our other PhD student, Hamida Palsy. And it's in nutrients. Again, we like nutrients, so if you want to have a look for the paper. Okay, so hang on a sec. Okay, so Dave's mentioned some things about eating behaviour, this cognitive restraint where people restrain their eating, and then that makes them more susceptible to binge eating later on. So although people in the short term eat less, sometimes in the longer term it can mean that they eat more. And cognitive restraint and uncontrolled eating, which is where you start eating and then have an eating episode and then carry on. So like you start on a packet of crisps and then you end up eating the whole packet of crisps. These are, and also emotional eating, which is people eating when they're upset or tired or distressed. These are all associated with increased BMI. And then food cravings for high calorie, high fat, high sugar fast foods are also associated with increased intake of these foods and also increased BMI. And BMI generally increases with age, although it doesn't always, but if you take people as a population in general, BMI increases with age. And food cravings and eating behaviours also change with age, and in general they go down a bit, but not necessarily in a predictable manner and not necessarily with everybody. So what about FTO? So FTO is a gene called fat mass and obesity-associated. And it was identified by what's called a genome-wide association study of GWAS, and it was the gene that was most associated with an increased BMI. So what you do is you take a huge population of people, like thousands and thousands of people, and you compare, you look at their whole genome and you say which genes or which gene variants are most associated with the characteristic you're looking at or the trait you're looking at, and in this case it was increased BMI, and this gene came out as having the variability in this gene was most associated with increased BMI. So that means some people have one at particular points in the gene. Some people have one variant, so this is quite really subtle changes. It's like one base change, like from an A to a T in that gene in your DNA, and just that small change can have an impact. And I'll explain a little bit how it works. So there's three. You could have three genotypes. You could be, so everyone who's watching this, I usually say everyone in the room, but we're not in the room. You can either have an AA genotype or an AT or a TT genotype, with A being the risk allele. So if you've got an AA or an AT, you're because obviously you've got two copies of each chromosome apart from the sex chromosomes. That's the version that's most associated with increased BMI. And 20% of the population are AA, 43% of the population are AT, and then the rest are TT. So you could almost say that the TT have a protective effect, and because the majority of people have at least one risk allele, and each copy of the risk allele increases BMI by 1.4 units, and the risk of obesity by 1.2 fold. So if you're an AA, then that you've got an increased risk of BMI by 2.8 units, which is quite a lot, really. And so this is the results from the G1 study. So you can see that the variance for FTO are all red dots, and this is what's called a Manhattan plot, because it looks a bit like the Manhattan skyline. And you've got all the chromosomes along the bottom, and then these like spikes or peaks are showing where there's genetic variability that's in this case associated with BMI. And so you can see that you get things lighting up as telling you which genes are associated with BMI or increased BMI. So you can see that there's other stuff going on, not apart from FTO. There's some other peaks around here, which are other genes. But then FTO is really a really high signal. Okay. And so what is FTO? What does it do? Well, it's something called an RNA methylase, which is an enzyme, and it doesn't really matter what an RNA demethylase is exactly. The important thing is that it's involved in the regulation of expression of other genes. So it controls or switches on or off expression of other genes. So it turns them up or turns them down, which means obviously a gene codes for protein. So that means you get more or less proteins. So if you're thinking about receptors, for example, and we particularly think that FTO might be involved in controlling leptin receptors, it means you might get more leptin receptors or fewer leptin receptors, and obviously that might have an impact on your appetite. Its highest expression is in the brain, in the hypothalamus, and we know that involves appetite. And if you modulate the levels of FTO in the arcuate nucleus in the hypothalamus, which obviously knows partly where appetite is controlled, that influences food intake. And knockout mice that don't have any FTO are hyperphagic. So the less FTO you have, the more you eat, and the AA allele is associated with lower levels of FTO. Okay, so here's our diagram that goes with what Dave showed us before, and we're probably looking around FTO having an effect on these POMC and NPY AGRP neurons, and it's probably regulating some of the genes that, and modulating the signal that's going on around here. Okay, but we don't fully know. Okay, so the question we asked was, does FTO have an effect by altering eating behaviours and food cravings? Now, this is a completely different type of study from the one Dave described to you. Dave had a nice, tidy research question. Okay, in this study we had 475 participants, but the reason we had so many was because this is a study that I run every year for my undergraduate project. So every year I have 2 or 3 undergraduate project students, and they each recruit about 20 or 30, maybe 40 people, and do a study where they use questionnaires, they measure eating behaviours, measure food cravings, measure exercise sometimes, but I'm not presenting that data today. And then they genotype for a gene, and they often genotype for FTO, but sometimes we pick other genes. And then with Hannah, our PhD student, what we did was we pulled all that data together. And I'd really recommend this as a way of those of you who are burdened down with teaching but have project students. I really recommend this method as a way of generating data, that you can get up to quite a large number of participants if you run the same project every year and they put the data together. The downside of it is that you don't get a full picture of what's really going on until you pull it all together, and then you think, I might have measured some other things actually, or done it a bit differently, but it's too late by then. Okay, so we had 475 participants, about equal numbers, male, female, average age of 30.7, but that hides a huge blob of people who were mostly students, and then a group of people who were quite a bit older that weren't. So actually there weren't that many people who were 30-ish. They were either sort of a bit younger or a bit older, and I think some people in this room took part in our study, so thank you very much for that, because we went around trying to get some people who weren't just students to so that the data wasn't so skewed to students. So lots of students and then an older group. Average BMI just over overweight, and we gave them 3-factor eating questionnaire and food cravings. We measured using the food cravings inventory, and then we genotyped for an FTO polymorphism, one of the ones that I showed you in the Manhattan plot. So this is the first few graphs I want to show you. So we're looking here at, does the FTO genotype, does it modulate the relationship between eating behaviours and BMI? So we've got eating behaviours on the y-axis, BMI on the x-axis. And then one of the first things I want you to notice is that the risk genotype, so that's the AA and the A2, is in red. And I don't know if you can see, but if you look at the BMIs, can you see how the red has a tendency to cluster over on the right hand side with the higher BMIs? So you can see this effect of FTO being associated with BMI being a little bit higher, but there's not a lot going on. Okay, the genotype's not really having an effect. So from these data, you could come to the conclusion that really FTO's effect on BMI is not being mediated by changing eating behaviour. So then we had a look at food cravings, and the food craving stuff's a bit more interesting. So here we've got different food cravings. We've got fast food cravings, and then sweet food cravings, and fatty food cravings. And here again, we've got the cravings up the side and the BMI along the bottom. And in this case, you can see that the relationship or the correlations are different depending on genotype. Okay, but this is when we kind of got into trying to interpret the data, because the fast food, the relationship between fast food cravings and BMI with the risk allele genotype is more pronounced, and there really isn't a relationship with the protective genotype, the TT genotype. So you could say, well, there's a positive in the people with the risk genotype, there's a positive relationship between fast food cravings and BMI. And so do the people with the A allele give into cravings more easily, and therefore that leads to the increased BMI if they've got increased cravings? But then that doesn't really match with what's going on with the fatty food over here, which kind of sees the opposite effect. And sweet food, we didn't really see anything. So is this by chance, or is there something different between fast food, which in the food cravings inventory is very much defined as, you know, kind of chips and burgers, and fatty food that you could possibly say is more involved in satiety and feeling full, you know, and not necessarily bad food? So we don't really know. And then we looked at, was there a difference if you split by sex? So on the left hand side you've got female, on the right hand side you've got male. Now I've got fatty food down the side, but we also, because I couldn't find a graph that Hannah plotted for fast food, because in the paper we actually present this in a table rather than pictures, but I thought pictures more interesting. But what I want to draw your attention to is the difference between the different genotypes, and that it's different for male and female. Okay, so the way it's sex specific. So so in female participants there's a stronger inverse relationship between food cravings and uncontrolled eating. Here, when you look at the relationship between food cravings and eating behaviour, in the TT genotype, the protective genotype, and it's not seen in the AA and AT genotypes. So maybe TT genotypes, maybe they're more able to modify their eating behaviour to control their cravings, to not give in to food cravings by eating in an uncontrolled way. But in males it's different, so I don't know if you can come to a conclusion about how that's different, why it's different in males, but I think the take-home message is that the relationship between these measures is different depending on sex, and that really needs to be taken into account. So in summary, FTO didn't influence the relationship between eating behaviours and BMI, but it does influence the relationship between cravings and, in our data, fast food and BMI. And it seems to influence the relationship between food cravings and eating behaviour in a sex-specific manner. And I haven't shown the data on this, but food cravings and eating behaviour in our data reduced with age while BMI increased, which kind of goes against what, when you're measuring acutely, and just it kind of goes against what I started by saying. But so if you take people and do food intake studies, you can show that the eating behaviour questionnaires relate to a higher food intake. But when you look at it over a long time, and a lot like people throughout their life and in different age groups, as BMI increases with age, but the food cravings and eating behaviours, particularly older people, tend to get lower. But there are so many things that might influence this change in how people perceive their body image, family circumstances. And this is why I think when we got to this point, we were thinking that we knew less than when we started out, almost. But anyway, the FTO did not mediate this this age effect we saw. So FTO is just one gene, and probably there's a whole load of others that kind of coming up in the sidelines behind FTO. So there's MC4R, which is important as well. So probably a polygenic approach where you look at several genes together might be better. But I think the key take-home message is that you really need to take into account age and sex and BMI when studying eating behaviour and food cravings, because the age and sex and when you're studying eating behaviours and food cravings, because male and female respond differently. These measures change with age, and also the influence of change in BMI on food cravings, which is in the paper, but I haven't really expanded on much here, is that as BMI changes, people's response by changing their food cravings or responding differently using their eating behaviour also changes. And which direction the relationship is, I don't think these, correlation is not causation. And so I think the take-home message is that you guys might be looking at more like Dave's kind of project with looking at appetite related to exercise, but don't forget all these other variables. They're really important as well. Otherwise, your data might be as confusing as ours. I think that's about it. Thanks.
So I didn't actually manage to unmute myself in time. So
oh God, I thought my volume had gone, and I'd be just speaking to myself.
Very good, very interesting talk. Thank you for that. It seems like a very complex problems. I was just intrigued with that the TT genotype response there with the gender specific response. I was just wondering what your next steps might be, or if you have any with that, what the research questions you might plan.
I think we need to kind of incorporate the sort of study that Dave showed with this, and do some feeding studies, and look at, so you'd have two groups, and you'd have the different genotype groups, and look at and then actually do things like ad libitum feeding, and actually see what happens in real life as well. And I think probably we definitely saw a difference in age, but I don't know if we didn't have a continuous age, it was just like two groups. And the thing is that because most of our younger people were students, I think even students are very, very different from people who are like 25, 26, 27. They're not, my son's a student, I'm going to say they're not normal. Oh, sorry, there's probably loads of students here. They behave differently. And so I think I'd try and do a wider range of people before.
It's always interesting, when you've got a very sort of large data set with a lot of different people in there. And I suppose until you step into that controlled research experiment where you can actually pose different questions, then you actually start to be able to get into a position where you can understand the mechanisms as to why, as opposed to just kind of what's going on. But it's very, very, very interesting.
I suppose we, like you say, we ended up with we think this might be going on, but we're not sure. We think this might be going on, but we're not sure. And testing those hypotheses is what we need to do next.
Fantastic. Do you have any questions from the floor at all?
Oop.
Okay. Okay. Well, great stuff. Well, thanks very much for that, Caroline. Very interesting to hear from you.